Okay, I think that we can start now. Good afternoon, everyone. Welcome to this ERN-EuroBloodNet topic on focus on inherited platelet function disorders. The first session is on platelet activation mechanisms by Professor Loredana. But before starting this webinar, I will just remind some housekeeping notes. So for these housekeeping notes, your microphone will remain muted during the presentation. Please note that this webinar is being recorded. You can submit your questions via the Q&A chat box at any time, and there will be a Q&A session after the presentation of Professor Buri. I want you to know that this program is accredited by the European Board of Accreditation in Hematology. You can earn 1 CME credit per webinar attended. You receive your CME credit if you attend the full webinar. You complete the feedback survey at the end that we will send you by email. You have an active IBA account, and you use your IBA email in the survey. So further instruction will follow after this event. And for this first session, I would like Professor Paul Saultier to take the floor and introduce this webinar series. Thank you, Professor Saultier.
Thank you, Christophe. And I would like to thank the EuroBloodNet team for helping us to set up this webinar series. So I'm Paul Saultier. I'm a pediatrician in Marseille University Hospital in France, and I lead the French Reference Center for Inherited Platelet Disorders. And I'm very pleased to welcome everyone to this first webinar. The main objective of this educational program is to disseminate up-to-date knowledge among hematologists, pediatricians, internists, pathologists, and all other healthcare professionals that are involved in the diagnosis and care of patients with inherited platelet function disorders. So this webinar comes after the former webinar who take place last year and who focused on inherited thrombocytopenia. So this year, the webinar series will focus on inherited platelet function disorders. And we know that these diseases are diverse, with diverse genetic backgrounds, with heterogeneous clinical manifestations, and the diagnosis of these diseases is often complex because of the difficulty with platelet function assessment, and there are overlap with acquired or secondary conditions. So we hope that this comprehensive series will cover both fundamental mechanisms of platelet activations, and this is the topic of today, and also state-of-the-art topics around diagnostic tools and current and emerging approaches for patients management. And so we are very pleased to start this webinar series, and we are honored that Dr. Loredana accepted to give this first talk. Loredana will introduce herself, but she is professor at the University of Perugia. And Loredana, the floor is yours. Thank you again.
Thank you, Paul, and thank you to all the organizers for this kind invitation. As you already said, yes, I'm from University of Perugia. I am a biologist with a PhD in cellular and molecular pathology, and I am currently associate professor at University of Perugia. I work in the group of Professor Paolo Gresele, and my research interest is focused on inherited platelet disorders, but also on platelet function and platelet physiology in general. So today this webinar is about platelet activation mechanisms. And I hope that I will give you a good introduction on the topic of this webinar series. I have no conflict of interest. Platelets are anucleate cells circulating in blood, and a normal platelet count is considered between 150 to 400 per 10 to the 9 liter. They are very small cells. Their diameter goes from 1.5 to 3 micron, and they have a life of about 8 to 10 days. And of course, they are mainly known for their primary function in hemostasis. In the blood of a healthy adult individual, we have a total platelet mass of 10 to the 12. This means that each day 10 to the 11 platelets are released from their bone marrow precursors megakaryocytes to maintain the normal circulating platelet count. Megakaryocytes differentiate from the hematopoietic stem cell, and several transcription factors that you can see in this slide participate in the maturation of the hematopoietic stem cell toward megakaryocyte. While in the stage of proplatelet formation, that is the release of long protrusions from the megakaryocytes from which the mature platelets are released, a major role is played by cytoskeletal proteins and also some platelet receptors. About the structures, platelets have a plasma membrane, like all the other cells, that is rich of receptors. They have a cytoskeleton that is mainly composed of actin and spectrin, and that supports the cell structure, but in the case of platelets is also very important for the processes of adhesion, for example, and activation. And they have a peripheral ring of microtubules that is essential for maintaining their discoid shape. Then they have organelles like the other cells except the nucleus. Therefore, there are mitochondria, lysosomes, peroxisomes, ribosomes, endoplasmic reticulum, and the Golgi apparatus. But there are also organelles that are typical of platelets. They are called alpha granules and delta granules. Alpha granules mainly store proteins that are released upon platelet activation. And delta granules or dense granules store nucleotides, ADP and ATP, serotonin and calcium. And finally, platelets have two peculiar membrane systems. One is the open canalicular system that is connected to the platelet surface and is crucial for the secretion of granules. It represents also a membrane storage to increase platelet surface during activation, because the platelets will change their shape during activation, and the membranes of the open canalicular system are able to increase platelet surface to take this peculiar shape. There is also a dense tubular system that is an internal smooth endoplasmic reticulum membrane system that regulates platelet activation because it sequesters or releases calcium, that we will see is very important, is crucial for the final platelet activation. Because this is the first webinar of a series focused on inherited platelet function disorders, I would like to mention when the effects of the proteins that I will describe during this webinar can cause an inherited platelet function disorder. In this case, we mentioned the transcription factors, and indeed, there are several inherited platelet disorders caused by mutations in the genes coding for all the transcription factors, or main of the transcription factors that I just mentioned, that are involved in the maturation of the megakaryocyte. And also there are defects caused by mutations in cytoskeletal proteins. Getting to the heart of our topic, that is platelet activation, platelets are, of course, involved in hemostasis. There is a platelet phase of hemostasis following the first phase, that is the vascular phase. During hemostasis, platelets undergo adhesion, release, and aggregation, and we will see all these processes in detail. And platelets also participate to the coagulation phase of hemostasis. This picture is a summary that shows all the platelet phase of hemostasis: adhesion, activation, and aggregation. We will see this slide again in this webinar, and now we will see all the steps in detail, starting from adhesion. So the platelet phase of hemostasis starts with platelet adhesion to the subendothelium that is exposed when a vessel wall is damaged. In particular, upon a damage to a vessel wall, collagen, that before the damage is in the subendothelium, is exposed to the blood flow. And this is caused simply by the injury that is made to the vessel wall. In plasma, there is von Willebrand factor, that is a big globular protein circulating in plasma. Von Willebrand factor has binding sites for collagen, and therefore it binds to the exposed collagen. But it has also binding sites for a platelet receptor that is called GP1B95. It is a complex composed of different subunits, and is the receptor, the platelet receptor for von Willebrand factor. The binding of von Willebrand factor to GP1B95 leads the platelet near to collagen and represents the very first step of the platelet phase of hemostasis. And now that the platelet is near collagen, thanks to GP1B alpha and von Willebrand factor interaction, there are two receptors on the platelet surface that are two collagen receptors, GP6 and alpha-2 beta-1, that can recognize collagen and trigger signaling, a signaling that we will see in detail in a few slides. And after the activation of these signaling pathways, we have the increase, we have platelet activation. That means increase of calcium inside the platelet, release of the granules, of platelet granules, alpha and delta granules, and release also of platelet agonists. Platelet agonists bind to other receptors that are present on the platelet surface, and these trigger other signaling pathways. Therefore, this is called amplification of the platelet activation. And finally, the amplification of platelet activation leads to alpha-2b beta-3 activation. Alpha-2b beta-3 is the receptor for fibrinogen, and fibrinogen is the molecule that can make a bridge between two different platelets. And this is platelet aggregation. But we will see this later, and now we come back to the activation. There are two main signaling pathways for platelet activation that are ITAM-mediated signaling pathways and G-protein-mediated signaling pathways. The main players of platelet activation are, of course, platelet receptors and their ligands that are called platelet agonists. Agonists are molecules that activate platelets after binding with their specific receptors, They can be released by granules like ADP that binds to P2Y12. They are produced by the coagulation cascade like thrombin that binds to PAR1 and PAR4. They can be produced by platelet phospholipids. This is the case of thromboxane A2 that binds to the TP receptors. Or like collagen, they are present on the damaged endothelium. Or they are circulating in plasma like epinephrine or adrenaline that binds to the alpha-2 adrenergic receptor. Going to the first signaling pathway, it's the ITAM-mediated activatory signaling. This is a pathway that is triggered by the ITAM sequences of specific receptors. ITAM means immunoreceptor tyrosine-based activation motif, and it is a motif present essentially on the Fc gamma R2A receptor and on the FcR gamma chain. Therefore, the ITAM-mediated signaling is strictly dependent on these two receptors. This signaling pathway involves Src family kinases such as Syk or adaptor proteins such as LAT. They all activate phospholipase C gamma 2, and its activation leads to activation of protein kinase C, and at the end to alpha-2b beta-3 activation. The ITAM-mediated activatory signaling is the signaling shared by GP1B95 and GP6. Therefore, it's the signaling that is immediately triggered after platelet adhesion to the exposed collagen. And let's see them on the detail. In the past, it was assumed that the GP1B95 interaction with von Willebrand factor provides only a means to tether platelets to the exposed subendothelium, like an anchor, without triggering any signaling. Now, we all know that GP1B95 mediates an intracellular signaling that leads to platelet activation and alpha-2b beta-3 activation. And in particular, it is mediated by Fc gamma R2A, the FcR gamma chain, but it may be also independent from these two molecules. About the signaling that is dependent on Fc gamma R2A and FcR gamma chain. It is a signaling that involves Src family kinases, Syk and Lin in particular. And as we already know, they activate phospholipase C gamma 2 that in turn leads to the production of the inositol triphosphate and diacylglycerol that lead to activation of protein kinase C and increase of calcium. This activates integrin alpha-2b beta-3. And about the independent pathway, it is a signaling triggered directly by GP1B95, and in particular because it activates this protein called 1433 zeta that activates Src, PI3 kinase, and therefore alpha-2b beta-3. About the signaling triggered by collagen receptors, some molecules are shared, like I previously said, with GP1B95 signaling. Also in this case, this pathway is dependent on FcR gamma chain, and so by its ITAM sequence. In this pathway, the interaction of GP6 with collagen activate lin and fin, that are two Src family kinases that then activate the item. The item activates Syk, and again we have adaptor molecules that lead to the activation of PLC gamma 2, formation of IP3 and diacylglycerol, activation of PKC, increase of calcium, and activation of alpha 2b beta 3. And of course, if genetic mutations affect the receptors for von Willebrand factor and for collagen, we have, as a consequence, an inherited platelet disorder. In the case of GP1B95, mutations in loss of function mutations in this receptor cause Bernard-Soulier syndrome. That is characterized by thrombocytopenia and defective adhesion, of course, of platelets, while gain of function variants cause platelet type von Willebrand disease, that is another platelet dysfunction that is associated with thrombocytopenia. And then there are also described cases of patients with mutations in GP6 and integrin alpha 2 beta 1. And going to the other pathway, the signaling pathway mediated by GPCR coupled receptors, receptors coupled to G proteins. This pathway is triggered by the interaction of ligands with platelet receptors that are coupled to G proteins, mainly G-alpha Q, G-alpha I, and G-alpha 12-13, but also G-zeta. About G-alpha Q, the activation of this G protein leads to activation of phospholipase C beta, formation again of IP3 and diacylglycerol, and therefore increase of calcium and activation of PKC that leads to integrin alpha 2b beta 3 activation. While G-alpha 12-13, its activation leads to activation of the Rho kinase that mediates the remodeling of the actin cytoskeleton of the platelet. And the activation of G-alpha I leads, on one hand, this is mediated by G-alpha I itself, downregulation of adenylyl cyclase, and as a consequence, a decrease of cyclic AMP. Cyclic AMP is an inhibitor of platelet activation, and therefore inhibition of adenylyl cyclase means platelet activation. On the other hand, the subunits beta and gamma of G-alpha I activate directly PI3 kinase that activates CalDAG-GEF1, that is a protein that leads to activation of RAP1, and RAP1 is a direct activator of alpha 2b beta 3. And in particular, the thrombin receptor, PAR1, and the thromboxane receptor, that is called the TP receptor, are able to activate all the three G proteins, while P2Y12 and its binding with ADP activate only the G-alpha I G protein. Epinephrine activates G-alpha I and also GZ that activates Rho. And of course, we have a platelet, an inherited platelet disorder, also if genetic variants affect the receptors for the G protein coupled receptors. Therefore, we have the TP receptor defect, the defect of P2Y12, P2Y1, while there are no defects described for the adrenaline receptor and the receptor for thrombin. And there are also defects of the enzymes that lead to the formation of thromboxane A2, because thromboxane A2 is a potent platelet activator that is produced by the platelet itself, starting from membrane phospholipids. In particular, there is an enzyme that is called phospholipase A2 that takes arachidonic acid from the platelet phospholipids. The enzyme cyclooxygenase 1 triggers the formation from arachidonic acid of prostaglandin G2, and another enzyme that is called thromboxane synthase transform PGG2 in thromboxane A2. And there are genetic defects described in thromboxane synthase, COX-1, and PLA2. They are more rare, but there are also genetic defects caused by mutations in G proteins, in other signaling proteins that I mentioned. I mentioned only TRPM7 that is involved in calcium increase, but there are also many other signaling molecules that I did not explain during this webinar, and that are mutated in inherited platelet disorders. And there are also disorders that involve directly calcium. This is the Storm-Morgan syndrome, that is a defect of the calcium channels of platelets. And we also mentioned platelet granules that are released during platelet activation that contains some of the platelet agonists. There are genetic disorders that involve alpha granules, genetic disorders that involve delta granules, and also combined defects of alpha and delta granules. Going back to platelet signaling, as we have seen, all the activation pathways that I just described leads to mobilization of calcium from their storages and activation of PKC. The elevation of calcium activates also CalDAG-GEF1, that is an activator of RAP1. And these recruits RIAM, that is an adductor molecule that facilitates the binding of two important proteins that are tallin and kindlin to the cytoplasmic tail of the beta-3 subunit of alpha-2b beta-3. The binding of tallin and kindlin to the cytoplasmic tail of alpha-2b beta-3 leads to activation of the receptor that, from a bent conformation, when it's not activated, takes an extended active conformation. This signaling that activates alpha-2b beta-3 is called inside-out signaling, because from the inside goes to the outside of the receptor that takes an active and extended conformation. The extended conformation is the conformation that is able to interact with ligands, and mainly with fibrinogen, that is the main ligand. And upon ligand of alpha-2b beta-3 with fibrinogen, that here is in yellow, alpha-2b beta-3 changes again conformation And another signaling is triggered that is called outside in, because from the outside goes inside the platelet. Tallin is involved, tensin, vinculin, because they bind to the beta subunit and link directly the integrin to the cytoskeleton. That means to actin. This binding promotes the cytoskeletal rearrangement that leads to platelet spreading, that is a firm adhesion to the damaged endothelium. And also to clot retraction, that is a movement of the platelet cytoskeleton that from outside goes inside and leads to the stabilization, to the firm stabilization of the clot. Meantime, SARC activates the focal adhesion kinase that reinforce this cytoskeletal rearrangement. So the outside-in signaling is the final signaling of platelet activation that leads to the stabilization of the clot. And perhaps you know this disorder, that is the most known platelet disorder, inherited platelet disorder, that is Glanzmann thrombasthenia. Glanzmann thrombasthenia is the genetic disorder caused by loss of function variants in the genes coding for integrin alpha-2b beta-3. In this disorder, the receptor for fibrinogen is not expressed or does not work properly, and this leads to the severe aggregation defect. However, there are also disorders of the alpha-2b beta-3 signaling that do not involve directly alpha-2b beta-3, but the signaling proteins that we just mentioned. There are mutations of CalDAG-GEF1 that lead to the disorder that is called CalDAG-related disorder. Mutations of kindlin that leads to a disease called LAD3 that is similar to Glanzmann thrombasthenia. And there is also another disorder that is caused by variants in the genes coding for alpha-2b beta-3. But in this case, we don't have loss of function variants, but gain of function variants. This disorder is called ITGA2B, ITGB3 related thrombocytopenia. And it can be considered like a variant form of Glanzmann thrombasthenia, where alpha-2b beta-3 is indeed expressed on platelets, and it is constitutively activated. Indeed, it is caused by gain of function variants. That means that the receptor takes a conformation that is constitutively activated. This is a work of our group where we demonstrated that in ITGA2B, ITGB3 related thrombocytopenia, a gain of function variants of alpha-2b beta-3 cause increased activation of the receptor. These experiments were made in CHO cells expressing the wild type or the mutant alpha-2b, or 4 different mutants of the alpha-2b beta-3 protein. And we have shown that the mutations lead to increased alpha-2b beta-3 activation. The increased activation causes increased internalization of the receptor. Indeed, in this disorder, there is a decrease of alpha-2b beta-3 on the surface, but not because the integrin is not produced, but because it is internalized, because essentially platelets tend to internalize the receptors that are activated and to recycle them. Moreover, activation of alpha-2b beta-3 leads to a permanent outside-in signaling, the signaling that I just described. That means increased phosphorylation of the focal adhesion kinase that as a consequence lead to a cytoskeleton that is always polymerized. This means that the platelets cannot mediate the last step, that is the step of clot retraction, because during clot retraction, the cytoskeleton that is polymerized retract, while in this disorder, we don't have the retraction. And this leads to platelet dysfunction, thrombocytopenia, because the cytoskeleton is very important for proplatelet formation. This is not the topic of this webinar, but an increased actin polymerization leads to macrothrombocytopenia, not only in this disorder, and the consequence is, of course, bleeding. Going back to the signaling, of course, there are also inhibitory signalings, such as DES2 that are mediated by G proteins. In particular, we have an inhibitory signaling that is mediated by prostaglandin I2, also called prostacyclin. This is a molecule that is released by the endothelium and is a potent vasodilator and an inhibitor of platelet activation. It binds to its receptor that is called the IP receptor, and this receptor is coupled to a G protein that is called G alpha S that activates adenylyl cyclase, that is the enzyme that mediates the formation of cyclic AMP. If you remember, cyclic AMP means platelet inhibition, because it activates protein kinase A that in turn downregulates a series of proteins that are important for platelet activation. Also, nitric oxide, this one, is produced by the endothelium. It is a potent vasodilator and inhibitor of platelet activation. Nitric oxide activates guanylyl cyclase that leads to the formation of cyclic GMP. Also, this means platelet inhibition, because cyclic GMP activates protein kinase G that, like protein kinase A, inhibit platelets by inhibiting a series of proteins important for activation. This is a work of our group. Here we have shown that not only the endothelial cells, but also platelets produce nitric oxides. Therefore, they regulate themselves, producing an inhibitor of platelets. And you can see these, for example, in this picture where platelets are in blue and nitric oxide is in red. In this experiment, thrombus formation was induced because platelets were allowed to flow under a surface covered by collagen. And when platelets meet collagen under flow conditions, we have the formation, of course, of a thrombus. And we have seen that during these, nitric oxide is produced. We can see, therefore, first that platelets produce nitric oxide, and that if we block the formation of nitric oxide with these LNMA, that is an analog of arginine, and therefore is able to block the enzyme that produces nitric oxide, that is called nitric oxide synthase. So if we block the production of nitric oxide, the thrombi are larger, and of course the platelets do not produce nitric oxide. Similarly, when we use the platelets derived from mice that were knocked out for nitric oxide synthase, and we performed the same experiment of thrombus formation under flow, here you can see that if nitric oxide is not present, like in this situation, thrombi are more and are larger. Therefore, platelets produce nitric oxide, and nitric oxide inhibits platelet thrombus formation. There is another regulatory mechanism of platelet signaling that is called platelet priming. In general, what does it mean? Priming. The priming is a regulatory mechanism that controls the response of excitable cells, like platelets, to stimuli. In particular, it means that the prior exposure to a given mediator can predispose the cell to a more effective response to a subsequent stimulus that is not a high stimulus, it is a subthreshold, a very mild stimulus. And in platelets, several physiologic substances can potentiate in this way platelet activation to primary agonists. What is important is that the platelet primers do not give full platelet activation, but only this subactivation that then potentiate activation when the platelet is exposed to a mild agonist. Let's see an example. These are other works from our group where we demonstrated that one platelet agonist is matrix metalloproteinase type 2. Like nitric oxide, also matrix metalloproteinase type 2 is released inside the platelets and is released upon activation. So again, it is a self-regulatory mechanism. Here we can see that upon exposure, upon platelet stimulation with thrombin, for example, matrix metalloproteinase type 2 is released by platelets, and this is dose dependent on the dose of thrombin. And here we can see that if we stimulate platelets with the very low dose of collagen, that in this case was 0.2 milligram ml, there is some platelet aggregation, but it's not a full aggregation. But if we expose platelets to MMP2 before activation with this low dose of collagen, the aggregation increases and is a full aggregation. And this demonstrated that matrix metalloproteinase 2 is indeed a platelet primer, and has an effect in increasing platelet aggregation. And we demonstrated it also in vivo in this model of femoral artery occlusion. In this model, we mimicked the same that I just explained, this mechanism of primer, because we produced two mild insults. The first is like the exposure of platelets to MMP2. So the first is the priming, and the second is a mild insult, so a subthreshold stimulus. And we observed that in this way, there is the formation of a thrombus. We measured the time to occlusion. And we also made this with mice that were knocked out for MMP2. And while in wild-type mice, after the second injury, not the first, the time to occlusion was decreased. In MMP2 knockout mice, this does not happen. This means that MMP2, again, is a platelet primer because we don't have this thrombus formation if the primer is not present in the circulation of the mouse and in the platelets of the mouse. We also showed the mechanism that modulates platelet signaling in response to MMP2. In particular, the receptor for MMP2 on platelets is PAR1, so the receptor for thrombin. And what happens is that MMP2 binds and cleaves this receptor in a position that is near the thrombin cleavage site, but is not the same. This activates PAR1, but while activation of PAR1 by thrombin, if you remember, activates all the three G proteins, GQ, G1213, and GI. When PAR1 is activated by MMP2 cleavage, the GI signaling is not triggered. This is why we don't have full aggregation, but only priming of platelets. This is a so-called biased signaling. That means that it is only partially, the signaling is only partially activated, in this case, because GI is not activated. And we also have evidences that the interaction of MMP2 with PAR1 is mediated by the alpha-2 beta-3 receptor. And finally, there is another regulatory mechanism of platelet activation. Indeed, platelets can synthesize new proteins when they are stimulated. Platelets do not have a nucleus, so they do not have DNA, but they have RNA, messenger RNA, that is inherited from their precursors megakaryocytes. Starting from this RNA, they can translate RNA in proteins and thus produce new proteins. Here on the left, you have some examples of protein that are synthesized by platelets upon stimulation, because there is also a constitutive protein synthesis in platelets, for example, the synthesis of actin or alpha-2 beta-3. In this case, we are here there are listed the proteins that are produced after stimulus. And platelets also have microRNAs that, as you probably know, are regulators of protein synthesis. And in particular, microRNAs stop protein synthesis because they bind to their target messenger RNA and they destroy the messenger RNA. Platelets have a lot of microRNAs and also all the mechanism for RNA interference, that is dicer, AGO2, and TRBP. And it was demonstrated by others that microRNAs regulates protein synthesis in platelets. But I would like to show you a very recent work of our group where we showed that platelets are able to neosynthesize dicer after stimulation with thrombin. Dicer is the enzyme that matures microRNAs. We focused on this, microRNA 223, that is a microRNA that regulates the expression of the ADP receptor P2Y12. And so in healthy people, the increase of dicer leads to increase of microRNA 223 and thus downregulation of the ADP receptor and reduced platelet reactivity. And then we demonstrated that in diabetic patients where the enzyme dicer is less present respect to healthy controls, and also in diabetic mice and mice knockout for dicer. Upon thrombostimulation, we have the neosynthesis of dicer, but it is decreased. Therefore, we have less microRNA 223. That means that we have more P2Y12 receptors, and this can explain the increased platelet reactivity of diabetic patients. Therefore, in this webinar, we have seen that platelet activation is orchestrated by ITAM-mediated receptors, by G-protein coupled receptors, and integrin alpha-2 beta-3 signaling, and that these canonical pathways regulate adhesion, secretion, and aggregation. However, platelets also have alternative regulatory mechanisms that are priming, neosynthesis of proteins, and the regulation mediated by microRNAs, and that many of these mechanisms, if dysregulated, can lead to disease. Thank you for your attention, and if you have questions, I'm happy to reply.
Thank you very much, Professor, for your thorough presentation. That's excellent. So as I reminded everyone in the chat, now is an excellent opportunity to ask Professor Berry questions directly here. If you are to put them in the chat, I can read them out loud for you.
So
Perhaps.
maybe while some questions emerge, thank you very much, Loredana, for this wonderful talk. Do you think there is a significant heterogeneity in the platelet population in vivo, and that maybe we can try to capture this heterogeneity with maybe new technologies or in the lab?
Thank you for your question. Indeed, there are evidences of heterogeneous platelet populations in circulations. For example, they differ for their RNA content. And therefore, with these new technologies, for example, single-cell sequencing, but also by flow cytometry, we can recognize platelets subpopulation that express some RNAs and not other RNAs, or some proteins, for example, tissue factor. There is only a subpopulation of proteins that express tissue factor and so that are procoagulant. This was already demonstrated. Or also the nitric oxide synthase. It is only a subpopulation. So yes, of course, there are procoagulant platelets, platelets that are more inhibitory. Yes, I am sure that in the future we will show that many different platelet populations play different roles. The role in inflammation, for example. Of course, I am sure that there are inflammatory platelets and other platelets that are more involved in aggregation and hemostasis, for example.
Oh, very interesting. Thank you. I don't know if there is a question in the chat. I don't see any for now. And also, what's your favorite platelet activation marker? If you do you use some platelet activation marker in the lab? You know, given that some assays will rely on platelets that are not previously activated, do you think there are some markers that are more efficient in capturing this?
You mean for diagnosis, for example, or
Yeah.
in general?
Yeah.
For example, for diagnosis, yes, we usually assess alpha-2 beta 3 activation with the PAC1 binding by flow cytometry. I believe it is an excellent marker, but not for early activation, but for full activation, of course. P-selectin is a marker of early activation because it is a marker for the release of alpha granules. They are the main activation markers that we test. Yes, essentially these two.
Thank you very much. It was really a great talk, very well illustrated also, with beautiful image that I think we can see in the book that you wrote.
Yes, there was a reference for this book chapter. It is not published yet, but it's the proofs are ready, as I know.
Wow, great. Thank you very much.
Perfect. If there's no more questions, perhaps I will make a quick announcement regarding the rest of the webinar series. We have one of the participants thanking you for your presentation. 1 second. So as Professor Paul Saultier pointed out, this is a very comprehensive webinar series, and we're delighted to be working once again, collaborating with Paul Saultier. I just want to point out that the next session is on November 12th, so in 1 month now, and you can actually use the QR codes that are on this PowerPoint to access the entire agenda, where you can also register for individual webinars or the entire series. So that's a great advantage of this. And I put together a quick QR code for the next webinar session, which is platelet aggregation, investigating a suspected platelet function disorder. And that's on the 12th of November, 2025. So just once again, I wanted to thank Professor Burry for her excellent presentation. I also want to thank Paul. It's a true pleasure to collaborate with you. And also I would like to thank our partnership with the CRPP, which is the French reference for Inherited Platelet Disorders, and also for the Filière MHEMO, which is the French Constitutional Bleeding Disorders Network. So to each and every one of you, thank you very much for joining tonight, and we look forward to seeing you at the next webinar, and we hope that you can join each and every session. So thank you once again for your time and your expertise and for your participation. Thank you, everyone.
Thank you, everyone.
Thank you, Gavin. Thank you, Loredana, and thank you, everyone, for joining.